Hello everyone, and welcome to the second half of our Blood Notes Chapter 19 lecture, where we last left off on Friday. We were talking about the origins of the formed elements of the blood, and when we say formed elements, remember we are referring to red blood cells, also known as erythrocytes, white blood cells known as leukocytes, and platelets, which are known as thrombocytes. We talked about how all of these formed elements, for the most part, except for the thrombocytes, well, even them a little bit, have to be underneath some sort of hormonal control in order for their production to take place. So hormones that are required are first the multi-colony stimulating factor needs to have an effect on the myeloid stem cells, and then erythropoietin, which is released from your kidneys, is going to direct the production of red blood cells. And then we had the granulocyte, monocyte, colony stimulating factors, and then we had monocyte colony stimulating factors and granulocyte colony stimulating factors as hormones or factors that are required to get the other classes of formed elements to be formed. We also talked about red blood cell recycling and how that process takes place as well. So if you're not familiar with that, I won't go over the entire lecture as a review this second, but if you're not familiar with that, please review the first Blood Notes lecture to go over some of those spots where you may have deficiency. So what we're going to do today is we're going to wrap up this lecture with a discussion in platelets and blood clotting. So the job of platelets is primarily for blood clotting. That's kind of their big role. They release clotting chemicals. There are various different factors that are released by platelets that will help to perpetuate a cascade of events that will allow for clotting to take place. The platelets aggregate together and they will clump up and actually patch temporarily the damaged blood vessel until it is permanently fixed with the fibrin and fibrinogen and the different connective tissues that make up the blood vessel. And then we also, with the patching of the blood vessel, because of this platelet agglutination, we have the function of a reduction in size of that break in the vessel wall, which makes sure that you don't, for the most part, bleed out uncontrollably. So the reduction in the size of the break in the vessel makes sure that you don't hemorrhage out too much. And keep in mind that sometimes there can be damages that happen to the body, that the clotting process will be unable to deal with. They might be too widespread, or the break in the blood vessel wall might be too large. And so that clotting, even though it's trying to take place, it's ineffective. So the way that we kind of look to make sure that your platelet counts are in their normal parameter is that we look at how many platelets we find per microliter. So about 150,000 to 500,000 per microliter, kind of like your normal range is in there. And once again, this just the factors that come into play are you a child? What age child are you? Are you a man? Are you a woman? So body size and type are going to have a factor to play in your blood volume levels, as well as red blood cell count, as we saw on our Friday discussion, and just the mass of your body. Thrombocytopenia, which means you have an abnormally low platelet count. If you don't have enough platelets, that can be problematic because it will not, when you do have a break in the blood vessel wall, you won't be able to have effective clotting. Thrombocytosis is an abnormally high platelet count, which even though you might think at first blush, well, isn't it a good thing that I have all these extra platelets floating around? But it actually can be problematic because you can have spontaneous clots that will form, and clots that form in pulmonary vessels or vessels of your heart and lungs. Clots in those places are not very good, and even if we have clots that form in the joints of your extremities, can be quite painful. So we want to make sure that we stay in our normal range for platelet numbers. Platelet production is called thrombocytopoiesis, just like we have erythropoiesis for the manufacturing of red blood cells. A thrombocyte is the fancy pants science name for a platelet. So thrombocytopoiesis is the fancy scientific name for the production of platelets. Platelets come from the same stem cell as the red blood cells, monocytes, neutrophils, eosinophils, and basophils. So everything except for the lymphoid cells, because they come from the lymphoid stem cell. The granddaddy of them all, if you'll remember, is a hemocytoblast, and that hemocytoblast differentiates into myeloid stem cells and lymphoid stem cells. That lymphoid stem cell is going to eventually, that myeloid stem cell that the platelets come from is going to eventually get bigger and bigger and bigger, and we would call that a megakaryocyte if it's under no hormonal control. So I'm going to bring this picture back up for you just as a reminder of what I'm talking about. So to make a red blood cell, we needed erythropoietin. To make any of the other white blood cells except for the lymphocytes, we needed this granules, the colony stimulating factors. But notice that to make the platelets, it was under no hormonal control. So if this myeloid stem cell is under no further hormonal control after the multicollineating stimulating factor is released, then it will just get really large and you'll have fragments of it break off. This really large cell, we call it a megakaryocyte, which will fragment into platelets. And let's get back to where we were. And it is the megakaryocytes that manufacture platelets from the cytoplasm of the cell. There is a hormone that's manufactured from the kidneys. It's not a colony stimulating factor in the same way that we saw that we had the the GM and the M colony stimulating factors that help to direct the production of the other white blood cells or other white blood cells, other formed elements. There is a hormone called thrombopoietin, which is found in the kidneys in the same place that we find erythropoietin. It helps to accelerate the formation of platelets. So normally the megakaryocyte is going to get large and fragment off and make the platelets themselves if they're under no other hormonal control, no other colony stimulating factors, no erythropoietin. But if the body recognizes that the platelet count is low and it needs to boost acceleration of that, then thrombopoietin can be released from the kidneys. Interleukin-6 will stimulate platelet formation as well. Once again, if there is a signal in the body that will say that, hey, we need more platelets, this hormone, this interleukin can be released in order to accelerate that process of platelet formation. And multi-colony stimulating factor, which we saw in the previous slide, that's where our myeloid stem cell is under the direction of that particular hormone. It's going to stimulate the production by promoting the formation and the growth of megakaryocytes. As long as that megakaryocyte or that myeloid stem cell that's now been under the direction of the multi-colony stimulating factor is there are no other hormones that are having an effect on that cell. It gets larger and larger and larger and becomes that megakaryocyte, which fragments off into our platelets. So the last part of our notes here we're going to talk about is blood clotting, or hemostasis. Hemostasis is cessation of bleeding, and there are 3 phases in this process. This is another great example of a short answer question. So the recycling of red blood cells, the formation of formed elements, and now hemostasis. Already I have 3 possible options in just this first chapter for possible short answer questions. So the 3 phases of it, first we're going to just look at the 3 phases, what they're called, and then we're going to talk about individually what happens in each phase. And for your short answer question, I'll probably ask you something to the effect of list and describe the events of the 3 phases that take place in hemostasis. So first we have the vascular phase, and when you see vascular, what probably comes to mind for you is blood vessels. So the vascular phase is going to be the activities that are happening with or directly connected to the blood vessels. Platelet phase is kind of intuitive, that we know that the platelets are going to be doing something or secreting something or having some sort of direct role in this phase. And in the coagulation phase, when you hear the term coagulation, you're probably thinking sticking together or agglutination. So that's pretty much what we're going to have happening there. So even though we haven't gone over the nitty-gritty and the specifics of what's happening in each one of these phases, we can probably have a pretty good idea of what we should expect to see. So our very first phase we have here is your vascular phase, which we've already decided must have something to do with the blood vessel. So in the vascular phase, there's been some sort of cut or injury that's going to trigger vascular spasms that are going to last about 30 minutes. Your blood vessels, believe it or not, are not just hollow tubes that stuff goes through. They actually have a layer of tissue, and they have a layer of muscle. So that smooth muscle is kind of the middle layer of it, and depending on what kind of blood vessel you are, you can have a lot of muscle that's there, or you can have very little. You can be very elastic, depending on what kind of blood vessel you are, as far as a vein, or artery, or a capillary, or you could be much more rigid, like arteries are much more rigid than what we see for veins. Veins are more floppy, but arteries have more elastic rebound. And we'll talk at length about this in the next chapter, the blood vessels chapter. So those vascular spasms that take place for 30 minutes, those are going to be the contraction of the smooth muscle layer of the blood vessel. There are 3 steps in the vascular phase. So for hemostasis, just as a review, we have 3 steps in that: vascular phase, platelet phase, coagulation. In the vascular phase, there are 3 very distinct portions to that. The first step of it is that the endothelial cells, or the epithelial tissue that lines the inside of the blood vessel, and I'm going to attempt to draw this, and bear with me. That's the outer layer of my blood vessel. Here is the middle layer of my blood vessel, and then here would be my squamous cells. So this is my endothelial layer. And that endothelial layer, if I was to blow it up, you would see that it's made out of simple squamous epithelial tissue. So just one layer of these amorphous type cells are going to make up that endothelial layer. So the endothelial cells are going to contract, and they expose the connective tissue, or the basement membrane, to the bloodstream. So I'm going to draw some donuts in here. These are red blood cells that are going to go through there. So these endothelial cells that we have there, that if I blew them up and drew them on the side, they kind of look like that. Those endothelial cells are going to contract because there's been an injury to the blood vessel wall, and they're going to expose the basement membrane to the blood vessel wall. So if you remember from AMP1, the basement membrane is the bottommost layer of the epithelial tissue, and it's going to connect it directly to the connective tissue. Those endothelial cells, once they have contracted and they've had their basement membrane exposed to those red blood cells, that's going to trigger them to release the chemical factors of ADP, adenosine diphosphate, tissue factors, and prostacyclin. So these chemical factors are going to stimulate the smooth muscle to contract, and it's going to also stimulate the other endothelials for cell division to take place. Because keep in mind, we do want to repair this blood vessel where it's had the injury or it's had the cut. So we have these chemical factors that will call for other cells to come to the area. You have ADP that's being released, so that if we just phosphorylate it or stick another phosphate group on it, we can have energy that we are requiring. So we're going to stimulate the smooth muscle contraction, A, and then B, we're also going to have the cells divide. Local hormones are going to also be released to call white blood cells to the area, and also to call platelets to the area, so that they can also release their chemicals. Endothelial plasma membranes become sticky, and because they're sticky now, those endothelial cells, those simple squamous epithelial cells, they're going to help to try to seal off some of the blood flow. Keep in mind, it's not going to be sufficient for the endothelial cells to become sticky themselves, so that blood vessel cells, when they go by them, they kind of stick to them to form a clot. You're still going to need to have other players in this. But in this very first stage of the vascular phase, this is kind of getting the ball rolling on trying to stop this bleeding from coming out of this blood vessel. First thing that we're going to have happen is that in the vascular phase, we have the endothelial cells are going to contract and expose the basement membrane. When they contract and expose the basement membrane, their chemical factors are going to be triggered to be released from those endothelial cells. Local hormones are also going to be released. All of these hormones, chemicals that are released, are done in effort to allow the muscular layer of the blood vessel to contract. It also calls for platelets and other cells, white blood cells, that can clean up any debris or bacteria that's gotten into the bloodstream, to clean up the area, and also stimulate the endothelial cells to start to divide and multiply, so that they can form new endothelial layer once that area is healed. And then the the endothelial membranes, those simple squamous epithelial cells that make up the inner layer of the blood vessel, those membranes are going to become nice and sticky to try to help seal off as much blood flow as possible. Next phase of hemostasis is your platelet phase. Begins about 15 seconds after the injury. This is where we're going to have the platelets adhere to one another. The attachments, we have platelet adhesion, and then we also have platelet aggregation, which means they not only attach to one another, they also are sticking together. So they're attaching to the endothelial layer of the blood vessel. They're also sticking together. So because of this adhesion and aggregation and the plasma membrane of the endothelial cells from the previous step, the vascular phase, because they're nice and sticky, that's going to form our platelet plug. And that helps to further close the break in the blood vessel. In the platelet phase, we also have certain compounds that are released. When we were in the vascular phase, we talked about how those endothelial cells release local hormones, ADP, tissue factor. The platelets are also going to release ADP. They're going to release serotonin and thromboxane A2. They're also going to release various different clotting factors. They'll also release platelet-derived growth factor and even calcium ions. So the platelet phase, the platelets themselves, not only do they stick together and help to form the plug, they're also going to release other chemicals to trigger more platelets to come to this area of injury infection, and also to cause for more cells to come to this area of injury and infection to help deal with the break in the blood vessel. If you remember way back in one of your first chapters in A&P 1, you probably talked about negative and positive feedback control. Negative feedback control, you said you're negating the stimulus, and the classic example that you may have used was body temperature, that if your body temperature gets too high, your body will do things to negate that stimulus, such as sweating, the dilation of blood vessels, etc. If your temperature gets too low, and it will do things to negate that situation as well, including the constriction of blood vessels pulling away from the surface of the skin and shivering. All of those things are done to negate the fact that the body temperature is too low to bring it back up. So that was an example of negative feedback. For the examples of positive feedback, we said positive feedback is going to accelerate events until we have some culminating response take place. And a good classic example of positive feedback, we said, was childbirth. You have contractions that get stronger and stronger and stronger until the culminating event or response is the birth of a child. And also blood clotting. Blood clotting is a positive feedback example, because you have the endothelial cells that are releasing compounds and tissue factors and hormones that will call for more platelets to come to the area, which then in turn triggers the platelets to release chemicals and clotting factors and growth factors and all other sorts of things that will call for more platelets to come to the area, which are going to release more of these clotting compounds until the culminating event is that we have a clot that's formed. Also, as kind of a bridgeway to what we've talked about last semester, calcium has a very important role in your body, right? So let's think of some of the things you require calcium for. Well, you require calcium in your bones. It helps to make your bones nice and hard as part of that calcium or hydroxyapatite that gives the bone its kind of strength. We needed calcium for synaptic transmissions in our nervous system. We also needed calcium could also act as a second messenger in the endocrine system. And then we see that calcium here is also one of the clotting compounds that can be released and required for proper hemostasis. So as we just illustrated, that blood clotting is a positive feedback loop, that you're going to keep getting all of these hormones and chemicals that are released to call more platelets, which are going to release more hormones and chemicals, which are going to call more platelets, and so forth. So we don't want our platelet plug to get too big, because that could completely cut off all circulation. So we're going to have some things that limit the growth of the platelet plug. One of those very important things are prostacyclin. So prostacyclin is released by the endothelial cells, the squamous epithelial cells that were found in our vascular phase that we talked about. It inhibits platelet aggregation so they don't stick together too much. You also have inhibitory compounds that are released by other white blood cells who have also been called to this area as a result of the various different chemicals that have been released by both the endothelial cells and the platelets. You have circulating enzymes in the plasma that help to kind of eat away at that platelet plug so it doesn't get too big, too out of control. And then when the serotonin is released from the platelets, which we saw in the previous slide, it has an inhibitory effect, or it negates the negative feedback. It's going to negate the over-agglutination, or the size of the platelet plug from getting too big. And we do this so that we can develop a blood clot that's being isolated in just this one area, and not one that just kind of is huge and it blocks off circulation to the area itself, and nothing can get through the blood vessel, or that gets so big and it breaks off and it goes someplace else in the body and gets stuck, in a smaller blood vessel and can be problematic. So here's a pretty picture of the platelet phase. So remember, in our platelet phase, it begins with the attachment of the platelets to those sticky epithelial surfaces. And as they become activated, when they stick to the surfaces of those epithelial cells, they're releasing these different clotting factors, ADP, platelet growth formation factor, calcium, serotonin, all of this stuff is going to be released. That's going to promote more vascular spasms, which are going to cause more platelets to come to the area, to cause more chemicals that will cause more platelets to come to the area, clotting, and then the vessel repair. So we had our cut, and now we have our platelets that are starting to form the beginning parts of our clot there. It's not completely done yet, because we have one more phase that we need to go through, and that's coagulation. In our coagulation phase, it begins about 30 seconds or more after the injury. This is the actual clotting, if you will, even though this whole process is a clotting thing that's happening. This is also a part of this cascade reactions, so these chain reactions of enzymes and proenzymes that are going to make sure that fibrinogen is turned into fibers that will, in essence, sew those platelets together to make sure that the platelet plug does not become dislodged from the break in the blood vessel wall and float somewhere else. We have 3 different pathways that will that we can use for this cascade reaction, and the culminating event that we want to happen from this cascade of events is to convert that fibrinogen, which is one of the plasma proteins that's in your plasma that we talked about in our previous lecture, into fibrin. And those fibrin molecules are what, as I said before, kind of thread all of those platelets together and keeps them stuck into the break in the vessel wall. So clotting factors are also called procoagulants. There are 11 different proteins and ions, such as calcium, that act as a clotting factor in the plasma. All of these clotting factors are required for normal clotting, and if you're missing any of them, or if they are malformed, then they could prevent hemostasis from taking place like it should. People that have hemophilia, one of their issues is that they are missing a clotting factor, so their blood does not, they have what people probably just generically call it bleeder's disease. If they have a bruise or they have a cut, it can be fatal for them because they will continue to bleed because their body doesn't have all of the clotting factors that are required for this last stage of hemostasis to take place. So the 3 coagulation pathways are extrinsic, intrinsic, and common. Extrinsic pathway is going to happen outside of the bloodstream in the vessel wall. The intrinsic pathway begins in the bloodstream with the activation of the proenzyme, and then the common pathway is just a combination of both the intrinsic and the extrinsic pathways. So it's kind of where they meet together, where the outside pathway meets the inside pathway. We call that the common pathway. So unlike extrinsic and intrinsic, there really isn't a location that this is happening in. It's more of a I don't know what the word I want to say is. It's more of a theoretical type of, or functional. So it's more of a functional grouping of things that are happening here. So the extrinsic pathway begins in the vessel wall outside of the bloodstream. Damaged cells are going to release this tissue factor, and remember the endothelial cells release tissue factor, and we have the platelet cells that are also releasing other factors. When we have the release of this tissue factor with other compounds like calcium, then it forms an enzyme complex, and it's going to activate this very special factor, factor 10. And I'll tell you why it's special in a second, because not only does the extrinsic pathway activate factor 10, but we also have the intrinsic pathway that is activating factor 10. So this is going to begin with the circulating proenzymes within the bloodstream. It's activated by enzymes. Its activation of enzymes are by collagen. Remember when we exposed that basement membrane and we had the basement membrane being exposed because of the contraction of the endothelial cells? And if you go back to A&P1, this is why a good understanding of A&P1 and Chapter 4 is vital, because we will keep talking about that throughout the semester. We recognize that there are collagen fibers in that connective tissue. So those collagen fibers that are in the connective tissue, they're going to help activate the enzymes for the intrinsic pathway. So the platelets release other factors, other clotting factors, and then it's going to also promote a series of reactions that will activate factor 10. Now that common pathway is where the intrinsic and the extrinsic converge. We need not only the extrinsic stuff to happen, but the intrinsic stuff to happen so that when they meet, they can form the enzyme prothrombinase. Remember, anything that ends in an ACE is an enzyme. So when they meet together, when we have the tissue factors plus the calcium that are meeting together, and we have this, let's go back here real quick just so we can look at it. We have the activation of factor 10, and then when we had our intrinsic pathway, the activations of enzymes and the platelets releasing their factors, kind of promoting the cascade of reactions that activates factor 10 as well. Then when they come together, when that factor 10 is activated on both ends of this functional thing that's happening of clotting, then we have the enzyme prothrombinase, which converts prothrombin to thrombin, and thrombin is going to be what converts thrombin to fibrinogen. Thrombin converts fibrinogen to fibrin. Fibrinogen is already kind of circulating around. Thrombin, we had to turn it into thrombin from prothrombin. Prothrombin is just kind of circulating around there, but we couldn't get it to turn into thrombin without the prothrombinase enzyme, which we didn't get until factor 10 was activated on both sides, both intrinsically and extrinsically. And here's a pretty picture of all this happening. So coagulation or blood clotting includes this whole cascade of events that leads to steps that will convert fibrinogen to fibrin. That's really the take-home message. So I'm going to put little stars on here that everything that we just talked about is to do one thing, really. We want to convert fibrinogen into these fibrous strands that will kind of thread through our platelet plug and make sure that it stays in the same spot. So as that fibrin network grows, the blood cells and the platelets get trapped in it, and they form a clot that seals off the damage of the blood vessel, and it also is going to keep that blood vessel or that platelet plug from dislodging and just floating through the bloodstream. Now, how we got to that was that we had our extrinsic pathways and we had our intrinsic pathways. Remember, extrinsic happens in the blood vessel wall, and we call them extrinsic. We really aren't dealing with things that are necessarily all brand spanking new, because these different tissue factors, calcium was already circulating, but the different tissue factors that are released from the damaged tissue that's in the blood vessel wall. So that's why we say the intrinsic pathway is going to take place in the blood vessel wall or associated with the outside of the bloodstream, because it's the damaged tissue that are releasing things like tissue factor 3, clotting factor 7, tissue factor complex, and so forth. So it's the damaged tissue that leads all of that to take place. The platelets that were circulating in the bloodstream, that's what we call the intrinsic pathway, because all of these chemicals that are released are found in the inside of the bloodstream. It's going to activate proenzymes, platelet factors, are released from the platelets. Calcium, once again from the bloodstream that was already kind of circulating around, goes into this pathway, and you have clotting factors 8, clotting factor 9, and they all are going to activate factor 10. Once factor 10 is activated, and we had this tissue factor complex along with the calcium that also activated factor 10, then that common pathway, which remember it's not an anatomical location, but because of the chemical convergence of the things that happen on the inside of the bloodstream through the intrinsic pathway and the outside of the bloodstream, the extrinsic pathway, it allowed for this enzyme, prothrombinase, to be made, which is going to convert this molecule, prothrombin, into a different molecule, or protein, thrombin. Thrombin is the chemical that is required to convert fibrinogen into fibers. What you need to remember about this coagulation phase, where does the extrinsic pathway start? Where does the extrinsic pathway start? Why is intrinsic called intrinsic? Because the factors that are released are coming from inside the bloodstream, and extrinsic because the factors that are released are coming from damaged tissues within the vessel wall. Why do they have to be activated? Well, we need to convert fibrinogen to fibrin. Know the role of prothrombinase. It's the enzyme that converts prothrombin into thrombin. What is the role of thrombin? Thrombin converts fibrinogen into fibrin. What are the role of the fibers? The fibers are meant to kind of string through or thread through those platelets so that they can agglutinate and become nice and secure and stuck in that area. Also understand that calcium is extremely important to this process as well, because it helps to make sure that the factor 10 is activated. And also know that factor 10 is important for the formation of this enzyme prothrombinase, and which is going to convert from prothrombin into thrombin and then fibrinogen into fibrin. So yes, it is quite a lot. It's quite a bunch going on there. But when I asked you the questions about hemostasis, you know the 3 stages: vascular, platelet, and coagulation. Be able to tell me what happens in the vascular phase, those 3 steps that took place in the vascular phase, what happens in the platelet phase, what happens in the coagulation phase with these 3 different pathways that are activated, and then it culminates with fibrinogen turning into fibrin. Once we get here, we have done our whole hemostasis process. So feedback control of blood clotting. It stimulates the formation of tissue factors, and we've already established before that blood clotting is a positive feedback loop. And what we mean by that is that as the platelets and the endothelial cells release more chemicals, and those chemicals are part of the intrinsic and the extrinsic pathway, and it's going to call for more platelets to come to that area and more of these chemicals to be released. And all of this does, all this positive feedback does, is to accelerate the clotting. As we also said before, we don't want the clot to get too big so that it completely blocks off circulation in the blood vessel. We want it to kind of just stay in the break in the blood vessel wall. And we definitely don't want it to break off and go somewhere else without dissolving. So you have plasma proteins, anticoagulants, antithrombin 3, which is going to prevent the conversion of prothrombin into thrombin, which then is going to prevent the conversion of fibrinogen into fiber. So it's just meant to keep all of those, that process from taking place. You have heparin that's released from basophils that are already in the bloodstream that will dissolve blood clots. And then aspirin, it's kind of like, you don't have aspirin that circulates in your body, but it also helps to chemically thin the blood out, if you will, and it keeps it from having these spontaneous clots. So heparin is something that you naturally have that is going to dissolve blood clots or keep the clotting process under control, and it's released from your basophils. So we understand the role of ions, calcium ion in this clotting process. So calcium ions were required for both the intrinsic and the extrinsic pathway as part of that cascade of events that culminates with the conversion of fibrinogen into fibrin. But also, we didn't talk about this a whole lot, but vitamin K is also essential for the clotting process to take place as well. Now in clot retraction, this is when the blood vessel is healed. We had enough endothelial cells to replace themselves, and we have healed the break in the blood vessel, so the residual bleeding is been reduced, and we've stabilized the injury site. Reduces the size of the damaged area, so it makes it easier for fibrocytes, smooth muscles, and other endothelial cells to complete the repair of the blood vessel. So we're starting to pull those edges together, and now the clot, we just need it as a temporary seal or Band-Aid, if you will. Now the clot can be removed from the blood vessel wall where the injury took place, because we're now repairing that more permanently. So with the development or the division of endothelial cells and smooth muscle cells, we're completing those repairs, and fibrocytes that will give range to a host of different things in the connective tissue that's behind the endothelial cells. We're trying to just make sure that this area is completely healed. So now that we have this clot that's no longer needed in this wall, we don't want it to just kind of float all around the place, because then it can get stuck somewhere. So we have what's called fibrinolysis, which is the process of dissolving the clot. So thrombin and tissue factors activators are going to activate plasminogen. When plasminogen is activated, it produces plasmin, and plasmin's job is to digest those fibrous strands, or literally to kind of deconstruct that clot. So remember, the fibrous strands were important for sewing all those platelets together. We're going to just sort of pull that whole thing apart now, so that we won't have this huge clot just kind of floating around the bloodstream and possibly getting stuck someplace that we don't want it to be. All right, so that is all of Chapter 19. On Wednesday, we will start Chapter 20, looking at heart anatomy.